It's a great pleasure to invite Volker Gerdts to present to us today. Volker is Associate Director over at VIDO. He's worked extensively with animal genomics and using genomics to develop a range of animal vaccines, some of which are commercial vaccines in the developed world, but actually some that are very important now for mobilizing into the developing world. And he's worked very closely with IDRC. We heard from Musa earlier on introducing this topic. And so it's a great pleasure to invite Volker here to talk about the development of livestock vaccines through the use of genomics.
Good afternoon, everyone, and thank you very much. Can you hear me okay? Does that work? All right. So as Dr. Mulroney said, I'm really going to bring a completely different aspect to what you've heard over the last 2 days. I'm going to focus on livestock, and the reason why we're doing this, livestock obviously is very important as well for global food security. And it also has many other implications, including, for example, getting out of poverty. So some of the vaccines that I'm going to talk about today really make a difference in some of these African countries for the small holders there. So typically you have 2 to 3 animals, and if you can ensure that those animals are doing well and you're making profit on them, then you can afford to have another animal and so on. And suddenly you get to the point where you're out of the or above the critical threshold, and so your family is out of that poverty range. So animal vaccines are not only good for providing protein for all of us in the future, but also to ensuring that especially the smallholder or the small families in these countries are getting to a level where they can have a sustainable income for their families. So as introduced, I'm the director of, or the research director of the Vaccine and Infectious Disease Organization. So you can see there, is there a pointer here? How does this work? So this is the building here on campus. So we're just across the street, if you want, from here. And you can see on there is, on the left side, there is our, what is called a level 2 building. So that's allowing us to work with all these common pathogens that we find around the world, that you find on every farm and so on. But then I want to put emphasis on this large building on the right side. And it's an architectural rendering. I didn't have a real picture on my computer. Oops. But what this is one of the largest high containment facilities in the world, right here in Saskatoon. And it allows us to work with so-called level 3 diseases. So those are diseases that potentially are deadly to both human and animals, so TB, HIV, and so on, but they don't have the large global or public impact like, for example, Ebola has. So they can be deadly, but they don't spread as much, as quickly as an Ebola would do, and so that's why they're categorized as level 3. And this whole building is designed to work with large animals. And so much of our research at VIDO is using novel technology, novel innovation, as the title said, genomics, for example, but now go into large animals and use large animals either as the target species, so we make vaccines for them directly, and I'm going to talk about 3 projects that we're currently working on with African partners, but also use these large animals as models for human diseases. And many of our research projects are actually focused on human diseases, but we're using these large animals since we're learning that large animals are much more predictable in terms of how the immune response works for humans than the traditional white lab mouse that you have. So let me just start here. These are some of the vaccines that the organization has developed over the last few years. But really, when we look into how vaccines have been developed for livestock over the last 2 centuries, much of it has been empirical so far. And so I don't even list those on there. So we make vaccines, and then we just inactivate them, and we inject them, and we see what happens. But with the rise of novel technologies like genomics, for example, we're now able to make vaccines that are very, very specific. So we can use vectors, bacterial or viral vectors, and then put a certain gene in there and then get a response to that particular gene. We can use infectious clones, so we make viruses ourselves, and then we can mutate them and then use them specifically as vaccine strains. We can attenuate vaccines through genomics, and we can obviously also make subunit vaccines so individual proteins express them, and then co-formulate them with things like adjuvants and so on to improve the response. But really, genomics has also offered us to do this, and this is reverse vaccinology, and this is an approach that I'm going to talk about in these 3 projects that I'm going to introduce today to you. This is now going essentially following the genomics concept, where you start just with the sequence, and then you express all these proteins, and you test them all in these animals, and you see protection, you see that these animals are protected, and then eventually you ask the question, well, what was that individual protein? So we're not doing what we used to do for many years, where you would have your protein, you work on it, you characterize it, you try to figure out what the function is. These days we're going back and we're just saying we're using all these proteins, we're just seeing which one works, and then later on we ask the question what it does. And then there's other things that genomics allow us to do, and for example, DIVA vaccines is one of those, and I just want to highlight this because this is a concept that maybe for plant biologists is not that familiar. So this is when we have now marker vaccines that allow us to vaccinate animals, and we can distinguish them by looking in the serum of these animals whether they have been vaccinated or infected. And by doing that, we can now ship them to other countries. So for example, when we had the so-called swine flu here in Alberta, it just broke out on a farm south of Lethbridge. It took less than half an hour for South Korea to close its border to Canadian pork. They said they don't want this disease in here, no more trade, no more shipping of animals. So the shipping of animals, the export, and now with Trump and all the changes that we're seeing, obviously politically that's very, very important to our markets. And so often we're hindered or limited by sending our animals to other countries because we can't say for sure that the animal is not a carrier for the pathogen. So if, for example, you have foot and mouth disease or something like that, everybody would say, keep your animals in your country, we don't want them, unless you can, through a test, prove that the animal had been vaccinated but wasn't infected. And so those are what's called DIVA vaccines, and you can see they're now the foundation for many eradication programs, Europe and so on. So here's an example of a pseudorabies. That's a virus in pigs, not so common here, although we have it. And then you can see there's these countries that are free for it, or they're monitoring for it, and then right on their border to the east is where the disease is at the moment. And so it allows you to essentially say this is a vaccinated animal, so we continue to trade with it, and this is an infected animal, we have to stop it. And so it's all based on what we're putting in the vaccine and how we then serologically can distinguish. But what I want to talk about today is an approach, and so I was asked to speak a bit about genomics and the technology, and that is called reverse vaccinology, and this is what I alluded to earlier. So here's the concept of reverse vaccinology. So you simply start with a whole genome of the pathogen of interest. And so using bioinformatics, and this is where you guys are really, really good at, we're just going in and we're looking at all the open reading frames, and then we're seeing where these potential antigens are, where are they located, and so on. But we don't really go and ask what are these all doing individually. We're just coming up with a list of antigens. And we're saying all of these open reading frames, we're just going to express them in E. coli or whatever model it is. And then in groups of 5, we just take all of these antigens, run down the list, we test them in animals, and then we see which one works. And so the key for this is that you have to have a good animal model that allows you to say, yes, we see protection against the disease. And if you do, then you can go later back in and dissect the individual antigens and then come up with your vaccine formulation. And so the in vivo testing is really the critical part of this. So you have your list of antigens shown here, and then you just group them in pools of 5 antigens, put them in the animals, see their immune response, expose them to the pathogen, see which groups are protected, then go back in, dissect those antigens in that group, and run them individually and see which one is protected. So it's a very simple approach, and the advantage of it is very, very fast, and it's also not very costly compared to the old approaches. And so the 3 diseases that I want to focus on today, and I spent most of the time on the first one, and then the other 2 is just a little, like showing you what we're doing in the animal health field. So the first one is the contagious bovine pleuropneumonia. That's a very important disease in Africa. It's one of the most important cattle diseases in Africa. There's vaccines for it out there, but they're not very good. They're not providing protection. And it's really one of these crippling factors in that it keeps, what I said, keeps these smallholder families limited to just a few animals. And so when they get this disease, it really affects their overall social health status. And then the other ones are the bovine TB. And I'm just mentioning this because a few years ago here in Saskatchewan and Alberta, we had a case of it. And I just want to show you what the financial impact of that is. And then another disease, Johne's disease, and I threw that in because it's an enteric disease. And it's quite interesting to understand. But the overall, what you see from all these pictures, these are chronic diseases, and you can see how poor these animals are doing. You can see the ribs. There's really not much meat there. They're poor doers overall and fade or waste away eventually. So here is CBPP. So this is the importance of this disease. You can see it's endemic in most parts of sub-Saharan Africa. The economic cost of it is estimated to easily be $300 million. It's probably underreported. And it also is important because it remains a threat to Southern Europe and the Middle East and potentially also to North America. It had been in North America a long time ago, but it's gone now. But there's always a chance that it could come back. So we partnered with friends and collaborators in Africa, in Kenya. So there was the Kenyan Agricultural Livestock Research Institute, CALRO, ILRI, the International Livestock Research Institute, and then also a vaccine manufacturer, which is called Kivavapi. And then also we had partners in South Africa on this and another project. And overall, this project was funded by IDRC, and I believe, Moose, I gave you this morning already a talk about this and maybe alluded to this. And so these are the current vaccines that are out there. You can see there is all kind of vaccines out there, live vaccines, killed vaccines, and subunit vaccines. They don't work really well and even have very severe side effects. So for example, some of these livestock live vaccines are known to have such severe side effect that when you inject it, the tail falls off. You can have necrosis in the tail, and the cow actually loses the tail. So that's how severe these side effects can be. Obviously, there is a need that we have better ones. And so this is the team just acknowledging all the people that are working on it. And so what we did is we took the genome of this mycoplasma. Mycoplasma mycoides is the pathogen that causes the disease. And then through a number of bioinformatical software and programs, we ran it through and eventually came up with what is a list of 66 antigens that we thought might be important to test. And so we were fortunate that at ILRI, another collaborator had already worked on many of them and had them already cloned. But eventually we came up with this list of 66 antigens and said, we need to test them all and see how well they work now as a vaccine candidate. And so there is a ranking that you have to do then to see how well they work. It's all bioinformatics combined with immune assays and so on. But eventually we then started to immunize these animals, and I'm just going to show you some of these data when you test now different antigens, and you can see how we do it. There's multiple trials. We have 10 groups in these trials, 5 proteins per this group, 10 animals per group, and so on. And then we go in and we just look at the immune response. And when you do that, then you can see that there is all your groups, and in blue is the antibody titer, and without going into detail, you can see clearly that many of these groups are responding well, and others are actually responding not so well, and that is to be expected from that. And so then we continued on with those, and we also looked at the cell-mediated immune response. So this is also another component, a very important component of the immune response. And when you do that, then you can see that we also got good cell-mediated immunity. And then the question is really, are they protected against infection? And so we're going in and we're using cattle that are found in Kenya. So there's the Boran cattle, and I'll show you later we're also using Zebu. We challenge them, and then really you're just looking at how well do they respond to the infection. And so there is really no bias at the moment. We're just running all these antigens through, seeing who does well and who does not well, and you can see when you then look at the protection, and this is just measured here and very simplified on looking at the lesion score, you can see that the various groups are inducing various levels of protection. There are some that there is essentially no protection, and others there is very good protection. And so this is then how we go about selecting the right groups and the right antigens and then we go in and decide these are the ones to use. We also look at isolation of these bacteria, these mycoplasma in their lungs, and again, the same thing you can see here. There's all your different groups, and you can see that for some groups, we actually didn't find any bacteria in the lungs, meaning that the immune response was very good. Whereas in others, for example, you saw there is still quite a few, so meaning the immune response induced by those proteins wasn't as good. So it's a very simple approach, and so then you pick just the groups that you want to go and continue with, and then look at some of the specific immune responses. And so without going too much into detail here, then we follow the immune response, how long does it last, how does it develop, and so on. And then we see, can we further optimize it? And so this is now where we then work on things like, can we make chimeric proteins, for example, to have these antigens expressed in a different conformation, and also maybe multiple antigens on the same protein? And can we formulate them now with adjuvants to improve the response? And so adjuvants are molecules that can help you to enhance the immune response to a vaccine and also direct it towards the type of immune response that you want. And so without really going into detail here, you can just see when you add these things, suddenly you can bring all your responses, which look like they're all over the map here, more tightly together and towards a certain type of immune response just by adding, for example, an adjuvant to it. And so this is then what we're doing. The second phase then is really to now use these adjuvants to improve the onset of the immune response, the duration of the immune response, the magnitude, and then also very critically, the type of the immune response. And that's something that is really important, whether it's a bacterial or viral antigen and so on. And so this is just showing you on the next slide here. I'll just jump forward. We now go in then and we test these different adjuvants here, and you can see that against the different antigens that we have down here. And so for each of these antigens, we're now looking at the specific immune response to just this antigen. And you can see that some of them are doing very well. Up here, my pointer is dying on me. You can see on top it's doing very well, and then on the first and second antigens, for example, there we have some lower responses. It's okay, it's just gonna, I can't hold it for a long time. And then the next question is really to see again, now from borons, that's one species that's affected with this disease. Do other species that are also affected with this disease, do they equally respond to this, do equally well? And so this is looking then in Zebu, and you can see the same thing here. When we go in now and we test our vaccine based on now 3 proteins only adjuvanted compared to a standard live vaccine that's out there at the moment, and then your control group, and then you look at the immune responses. This is very complicated, but you can see again just how they differ to each of these antigens to essentially look at what happens after you infect them. And so this is now looking at how sick are these cows getting after you infect them. And so what we do typically is we measure the body temperature of these animals, meaning if they develop a fever, then they're getting obviously sick from it. And you can see in our control group, these animals are developing fever. They're above this line here. Whereas in our vaccinated group, for example, none of these cows actually develop fever. So you can see clinically then they're protected against it. And then when we look at the pathology, so opening them up at the end of the trial and see if we can find any lesions, then you can see, and this is just simplifying it here, in the vaccinated group, there's only 1 out of 8 animals that even showed some clinical scores or pathology scores, and you can see scores very low compared to the other 2 groups. Here's the standard vaccine that's out there at the moment. 5 out of 8 animals were still getting sick, and you still see pathology then compared to this new vaccine, whereas there's really only 1 and very low pathology scores. So it just shows you how you can just use genomics to essentially come up with this very simple approach, test them all, then limit it to a few only, and eventually formulate it with a good adjuvant. And if you're lucky, then it works as well as this does. So the vaccine then was licensed to a Kenyan vaccine manufacturer, the KVVAPI, the Kenyan Veterinary Vaccine Production Institute. And we're starting now a field trial next year in 2019 in a couple of thousand animals out there to see how well it works. And we're already working with the regulators on getting approval for this so that it can be then eventually used in African countries. And so current activities are focused on or are using this large scale now so that we can manufacture the vaccine and get all those things optimized so that we're then ready to produce the vaccine and eventually for KVVAPI to sell the vaccine in Africa. Now the two other examples, I'm just going to have 2 or 3 slides each, not very much, but it's another important disease that I just wanted to highlight, so bovine tuberculosis. It's a disease that actually is not really relevant in cattle anymore. It's a regulatory disease, it's a trade disease. And so we don't really see it that much in our cattle populations. There's countries in the world, though, that have it. But when we see it, it can have huge implications on the Canadian industry, for example, or other national industries. And so this is an outbreak that happened in 1617 in southern Alberta and Saskatchewan. It was initially 6 animals that were infected, and it resulted in over 50 ranches with over 40,000 animals that were quarantined afterwards. And after 12 months, there was about almost 12,000 animals destroyed and more than 30,000 animals which eventually were released from the quarantine. And the economic impact was massive for this country. It was millions and millions and millions of dollars. And it was clear that what was needed is better diagnostics, but also better vaccines for this disease. And so as you know, there is BCG, although we don't use it anymore. But the issue with this disease, as I alluded before, is that we don't use vaccines because we can't treat them anymore. Nobody wants these cows that look like they were infected with bovine TB. So the future vaccine, the vaccine that you could use for this in the case of an outbreak or even maybe preventatively in your whole population, needs to be a DIVA vaccine, something that allows you to distinguish between infected and vaccinated animals. And so again, the same idea here, we're running through the same process. Now it's much larger mycobacteria. There is many more antigens than in these mycoplasma. But the same idea, we're running through it, come up with this list, we're testing them, and eventually derive to a vaccine. But the nice thing is, with the concept of having a DIVA or the need for a DIVA vaccine, we're starting right up front saying whatever we're using here on our list, whatever each of these individual proteins we're using, it needs to have the potential to be a DIVA marker eventually. So it needs to be something that we can later on say this has been vaccinated and not from an infected animal. And so this is just, you can see we made, in this case, we cloned 111 antigens. The number is even higher now. We expressed most of them right now. We purified them and tested them already. And this is a project that is funded by Genome Canada and Genome Prairie and Genome BC and even the provincial government here in Saskatchewan through ADF. And so we're now running them all through large trials in our high containment facility. Same idea, but this is now level 3, and this is a very complex disease. But the same idea, we're testing them all now, and we're looking for pathology. So when you go in after your trial and you look, do you see these typical TB lesions? And you can see that these lymph nodes, and here overall there's this severe lesions, this CASI inflammation, this necrotic tissues here, typical of TB. And so then the same idea is going through and seeing which one works well and which one doesn't. And so the last disease is just because it's an interesting model. It just shows you how we're using these animals these days. So this is Johne's disease. It's an enteric disease, and it's a huge problem around the world. It affects essentially every dairy cow or every second dairy cow on this planet. It's a disease that eventually leads to chronic wasting of these animals by essentially causing enteric problems, chronic enteric problems. And so the model here we are using is not testing them in animals, but we actually have a surgical model that allows us to go into the animal, into the intestinal tract, and do what is called a gut loop. So we surgically make a independent loop here, and then we put our pathogens in there, and we see how well the vaccine protects against it. So it's just another example of how we do vaccine research using these large animals now. We're infecting them, or we sometimes only use surgical models like that. Now, when you go in surgically into these tissues, now you can also ask the questions, what's the transcriptome of all these sites, whether it's individual cells, so individual monocytes, for example, or these infected gut loops. And you can see there's a complex process which is probably similar to what you guys are using in the plant field, I'm sure. But eventually you derive down here with the cDNA libraries, and then you can essentially look at the transcriptome in these animals. And so this is just showing you, even for yonis, we're now currently over 120 antigens that we're using there for our vaccine candidates, and we're now running them through these gut loops, trying to come up with the same idea that we did for the CBPP for a vaccine that protects against this disease. So to summarize, genomics these days offer a great tool to design modern vaccines for livestock. And I talked about this reverse vaccinology approach. It's a fast and unbiased approach to identify protective vaccines antigens. And so it's very fast, novel, and it's not very costly. And we've used it now for these 3 novel vaccines in cattle that I talked about today. But we're also using the same approach then for swine and poultry and others. So with that, I'd just like to acknowledge all the people that were involved in the work, and I'd be happy to answer any questions if you have. Thank you very much.